Next is oral presentation number 26, Toronto, Canada, Ryerson University, their Mission 8 flight experiment with undergraduate sophomores and 11th graders.
Good afternoon, faculty, students, and organizers. My name is Pri Kaelan, and today Francis, Burgess, and I will be going over the technical details of our presentation. Now, before I continue, I'd like to acknowledge our group members who weren't able to make it here today. We have Gemma, Kuganthini, and Maudlin. And something interesting that we did at Ryerson University is that we partnered up with David and Mary Thompson Collegiate Institute to mentor these students through the scientific process as we collaborated with them. We are delighted to have our experiment selected for the Student Spaceflight Experiment Program. Our experimental proposal is to test whether microgravity will affect the growth of Pleurotus ostreatus. So, why mushrooms? Well, Pleurotus ostreatus is a unique species of mushrooms that is commonly known as oyster mushrooms. And it is the second most grown edible species of mushrooms. And rightfully so, because it has 3.5 grams of protein, 5.1 grams of carbohydrates, and 3.5 grams of fiber per 100 grams. So, if grown successfully, it can be an excellent food source for the crew on flight. So, additionally, it grows under ideal temperatures ranging from 10 degrees to 35 degrees Celsius. And aboard the ISS, the temperature ranges from 21 to 25 degrees Celsius. The growth capacity of Pleurotus ostreatus allows for the use of many different substrates, such as coffee pulp, paper waste, and degrading vegetables, all of which can potentially be available on flight, especially when we're considering long-term flight missions. There have been 2 prior experiments done with different species of fungus, both of which have shown great success in microgravity. So we do look forward to seeing how our results will compare to theirs. So from our data, we'll be answering the question, will microgravity affect the growth of Pleurotus ostreatus? Our hypothesis is that microgravity will affect the growth of Pleurotus ostreatus. So some of you in the audience may be wondering, what's so unique about mushrooms? And what exactly will be growing in our tube? Well, mycelia is composed of hyphae. So a great analogy to really understand what's going on here is to know that roots are to plants as mycelia are to fungus. So essentially, the mycelia will be growing on the growth substrate, and it's going to be growing roots, which are called hyphae. And it's these hyphae that will be extracting the nutrients from the growth substrate to allow the fungus to grow. Now, I'm going to hand it on to Francis to explain the experimental procedure.
Our experiment will be using the Type 3 FME, and the 3 volumes will be filled accordingly. The first volume of our tube will be filled with the pearl oyster mushroom spores. The second volume will be filled with the rice straw and the water gel crystal mixture. The third volume will be filled with the formalin solution. All of the materials will be sterilized to prevent any growth of unwanted organisms. Interactions with the tube will go as follows. On the 14th day before undock, clamp 1 will be unclamped, and the tube will be shaken vigorously for 30 seconds to mix volumes 1 and 2. This will activate the spores and stop the growth. After 9 days, on the 5th day before undock, clamp 2 will be unclamped, and the tube will be shaken for another 30 seconds vigorously to mix all 3 volumes. This will stop the growth and preserve the mycelia that have grown. Mycelial content and physical differences will be the basis of analysis of our experiment. Mycelial content will be quantified by measuring the differences in concentration of glucosamine. Mycelia is made up of chitin, which gives it strength and integrity. Chitin gets its strength from the cross-linking of the sugar N-acetylglucosamine. This sugar is our indicator of growth. The only source of this sugar will be the mycelia of the fungus. The mixture in the tube will be washed with sterilized distilled water to get rid of the remaining rice straw, formalin solution, and water gel crystals. The mycelial content will be collected and dried, and this content will be weighed to obtain the dry biomass. A sample of the dry mass will undergo microscopy analysis to look for physical differences. We are interested in finding differences in color, density, diameter, and growth patterns of the hyphae. The remaining bulk of of the mycelial content will be assayed to break the sugar cross linkages. The solution will undergo chromatography to separate out the glucosamine. Colorimetric measurements will determine the difference in the glucosamine concentration. Any difference in color will mean a difference in concentration of glucosamine. We would like to thank Dr. Brian Koivisto and Nathan Battersby for facilitating SSEP at our university and making this opportunity possible. We'd like to thank Sandra Solomon for making this program happen and securing our sponsors. We would like to thank Dr. Emily Agard and Lindsay Jackson for sharing this opportunity with the Toronto School Boards and Let's Talk Science. We'd also like to thank Graham Pearson, our collaborators Mira Jabarov, and Dr. Joseph McPhee for their advising that helped shape our experiment. We'd like to thank our sponsors Bombardier, Magellan, Ontario Ministry of Research and Innovation, and NSERC. We'd also like to thank Ryerson University and Let's Talk Science. Last but not least, we'd like to thank Dr. Jeff Goldstein for getting our community involved in such a short timeline. Thank you to the rest of the SSEP team and to the audience for listening to our presentation.
Okay, this is another first, another notable team. First of all, the second Canadian team to participate at the conference. And my understanding is that the five researchers that make up your team, 3 of them are from Ryerson University, are undergraduates, and 2 of them are from Thompson and Mary Collegiate, is that right? High school?
Yes.
And so this is the first presentation by undergraduates at the SSEP conference. And I think that this, right after fourth graders that we just heard, really is a testament to how broad a range this program addresses across the pipeline in education. So it's wonderful for you all to be here, and thank you for being the first undergraduate presentation of the SSEP National Conference.
Thank you.
Do we have any questions for this team?
Hi, I'm Maddie. I'm from Toronto. I was just wondering if you could expand on your hypothesis and in which way you were expecting the microgravity to affect the growth of the mushrooms?
So in our hypothesis, we indicated that we expect a different growth. There have been experiments where the color of the hyphae and the sizes of the hyphae themselves are different. This environment is extremely unfavorable to the fungus, so we'd like to see if we'd grow less or more. To be more specific, we hypothesize that we will grow less mycelia in the tubes itself.
Thank you.
Thank you.